Hello, everyone. Thank you for joining me today. My name is Edwin Solares. I am a postdoc at the Avocado Lab at the University of California, Davis. My contact information is below. Today, I'll be talking to you about Persea americana, commonly known as avocados. We're going to discuss the origin, history, population structure, and heterodichogamy, which is basically just the flowering avocados, which is actually pretty cool. Our work is available as a preprint in bioRxiv. So why avocados? Well, agriculturally they're important for United States, Latin America, and Africa. Also, very little genomic tools have been made available, and these are necessary for improving breeding. Why should we improve breeding? Well, they require a lot of water. It's also led to armed and water right conflicts in Latin America. And using these genomic tools, we hope that breeders can improve on avocados, improve their drought resistance and disease resistance. There also exists high diversity in phenotypic variation, which we'll see on the following slide, and the origin is poorly understood. There has been previous analysis that has shown three separate domestications events, and also they have very full flowering that is very basal-like. Here are some examples of fruit diversity alone in Mexico only. So we're still actually missing some of the diversity that's in Central America. If you look at Mexico here, we can see that there actually are a lot of Persea taxa, and we have areas of high density here, and those actually overlap with areas where avocados actually grow in the wild. The three separate domestication events are believed to happen in the Mexican highlands, the Guatemalan highlands, and the coastal lowlands. Now, the Mexican highlands are actually a lot colder and drier than the Guatemalan highlands. Here's an example of the Mexican avocado. The fruit tends to be smaller and thin skin, whereas the Guatemalan tends to be a lot bigger, round, and thicker skin. And also, the lowland avocado previously has been called like Antillean avocados and West Indian avocados. Actually, not descriptive of where the avocados come from, so we're going to call them lowland for the purpose of this talk and the study. So, some of the questions: how different are avocados in Mesoamerica genetically? What is their origin? And does it have a similar situation as teosinte in maize? And there's also two schools of thoughts where there's a domestication event similar to that of teosinte in maize, but also possibility of there being a progenitor that has now gone extinct and is represented by the three racial ecotypes. What is that divergence time between the three botanical races, and can we infer a population structure based on our data analysis? Has integration played a role in domestication, also similar to in maize? We saw a lot of overlap between the Persea species, giving rise to opportunities for integration. And can we identify the genetic basis for flowering, and are structural variants involved? Those are areas of the genome where we have genomic windows that have been either inverted, copy paste, or cut and paste, or deletion, meaning it's just removed from the genome. In order to answer these questions, we have to one, generate a genome assembly. We also need to resequence avocado cultivars. We also imported data that was publicly available from a previous study, Redon et al. And PNAS, that was published in 2019. We want to identify those mutations and do a comparative analysis using that. And we're looking at, I'm interested in looking at SNPs, which are single point mutations in the genome, and also indels, which are some of those copy, paste, cut, paste, and deletion situations. For that, we need good sampling across the three botanical races, but as well as the flowering types. We want to reconstruct the population structure and look at the integration. In order to do that, we're going to look at the PCA admixture and phylogeny. For purpose of our talk, we're going to look at PCA since the admixture and phylogeny results are identical and basically have no new information. We'll also look at possible domestication by looking at divergence time between the three racial ecotypes. And in order to do that, we have to identify good outgroups and computationally estimate our population size over time using MSMC to investigate the flowering for heterodichogamy by doing FST analysis between the two flowering types. Avocado genome. I produced a conjugate assembly and a scaffolding assembly. Avocado genome expected size about 900 megabases and consists of 12 chromosomes. 30% of our genome was gene coding, and 60% were by repeats. I ran my a program that I published in 2021 in bioinformatics in order to reduce the genomic noise and make a pseudo-haploid assembly, as there was alternate contents in that previous assembly. We took the top 12 fragments at 70% of the genome. The scaffolded genome was used in order to calculate this also. the top 12 fragments for 78%. We scaffolded using a genetic map. I took those 12 pseudomolecules and took the in-place contigs, which are contigs that are in the haploid genome but not in the scaffolded genome, and created a new genome that was 1 gigabase in size. And here's haploid in action, firing its QPs at Avocado, and that work was published in BSC Bioinformatics in 2021. If you look at sample origins, we do have samples that were in Mesoamerica, and we have the sampling information, but for a lot of samples in California, Florida, Israel, and Australia, we didn't. So we had to infer based on historical classification, and we actually got to reclassify some of the Mexican avocados as hybrids. Here we have PCA of Persea with Persea shediana. The only reason is because there's been previous research that has shown a different individual, Persea shediana, that clustered close to the Mexican avocados, and you can see that here. This is what shediana looks like. It actually is similar, very similar to avocado, and is sold in markets across Mesoamerica. If you look at the PCA, for avocados, we see a group clustering here, which represents Guatemalan, lowland, and Mexican avocados here. When we look at divergence time, we use MSNC2. And in the y-axis, we have the effective population size of Mexican, lowland, and Guatemalan pairwise comparisons. And x-axis, we have time. When we look at the time scale of domestication of maize, that will give approximately the line here. We can see that that's pretty far from these dashed lines here. Those dashed lines are actually the most likely point in time when these populations diverge. For example, here we have the lowland Mexican, Guatemalan Mexican diverging 1 million years ago, and the Guatemalan lowland diverging 40,000 years ago approximately. Here we can see the populations tracking, basically meaning that gene flow was active between the two populations, but then breaking down and having to convert divergence between the populations there. So actually, avocados have a basal like flowering. This is evident because if we look at different species that are native to Japan and Southeast Asia, the flowers morphology is almost identical, and as well as their behavior. So let's look at that behavior for flowering. These are some of the pictures I took of our own avocados. So flowers in avocado, they start out female in their first phase and male in their second phase, and this is what that looks like here. There are two types, the A and B type. Flowering occurs over 2 days, with the A type female in the morning closing up and opening back up male in the afternoon of the following day, whereas the B type opens up female in the afternoon of the first day, it closes up and opens up male the following day. When we look at the flowering those pictures that I had earlier, here we can see that those are identified here for the stamens and the pistil. Avocados are mostly outcrossing. Very little selfing does occur. It does occur in like special situations, but mostly it's just outcrossing. They produce millions of flowers, but only retain a little bit, about 0.1%. Here we have the FST analysis for type A and B. We have the genomic regions here. We did the genome scan with 20 kilobase regions windows, and we have the mean FST here, 1.0 being the most divergent and 0 being indistinguishable between the two groups of A and B. If you have something like this where the dots are tracked on this line here, we would say that chromosome 2 is indistinguishable between the types. And if we had dots that track this line here, we would say not only is it divergent here, but also a buildup, meaning a possible selective sweep. When we actually look at the data, we have multiple points that are possible selective sweeps. Those are chromosome 1, 3, 6, and 10. When we look at the genes in the top 1% of windows, we're actually being very conservative and not just looking at, like, say, the top 5%, but instead of the top 1%. We have genes in some of those chromosomes, chromosome 1, 3, and 10, and also 5. The cool thing about these genes is actually related to flowering. For example, floral identity, photoperiodic flowering, circadian rhythms, and also the release of volatile compounds in flowers, which are probably most likely helpful for pollination. We also have several genes in unplaced contigs, meaning our scaffold is not complete. More scaffolding would be required in order to place these contigs somewhere. Most likely, these would probably be in something like a sex chromosome, since we have flowering types about 50-50 within the progeny of a tree. In 2019, I was a co-author in a publication where we looked at grape color um, in Chardonnay and another variety of grape. And when we compared the two genomes, we saw that there was actually a inversion there. If they were identical, we would have something like this, but instead we see this inversion here. And this is important because it is my hypothesis that I hypothesize that SVs are probably inducing some of the genes that are causing these differences in flowering. And there are other SV-induced phenotypes, for example, in blood oranges, the reddening in blood oranges, branching in maize, and pigmentation in snapdragons. Well, this particular example is a SPA1 gene homolog from those cool genes that we saw earlier. Here we have a genomic window of about 20 kilobases, and with the FST window here at the edge. When we look at the gene at this genomic region, we see in green the gene coding regions responsible for making the protein. Here we look at the repeats to see if they overlap with any of those exons, and they don't, actually in non-coding regions. I then took the type A and type B Illumina reads and stacked them up to see what was going on, if there was any differences or if we saw any structural variants. And when I did that, we can see that at the beginning of that gene, we have uniform coverage with some drops in T's, which is kind of as expected, but we saw these, or I saw these breaks here in coverage for the type B, and then type A, it was near complete coverage across that area. So if we zoom into these regions, we can see that they're in repeat regions, but there's this exon here, which looks like it's a deletion in the type B. I then want to look at coverage across all genes, and here we have the type A avocados from 0 to 1 in coverage, and normalized to 0 and 1 in coverage for type B. If they're identical, we should see dots on this diagonal. If there's a type B bias, we would see a regression line to the left, and if there was type A, we would see a regression line to the right. When we look at all genes, especially the bottom 99%, meaning genes not in those top 1% FST windows, we can see that we do have a type A bias in those, but when we look at the top 1%, the genes in the top 1% FST windows, we see even a further bias there. And that SPA1 homolog is actually here, and then this lost one, and a lot of those genes are actually in these regions where they're type A bias, which is pretty cool. So there is probably some kind of structural variation in these flowers, and all these genes with flowering implications. So how different are avocados in Mesoamerica? Actually quite different. It's in the PCA they clearly classifying PC space. We have the three separate clusters, which were pretty far apart. As far as the origin, it was completely different than what we expected. We saw that the divergence time was not in human timescales, but in geological timescales. So we still have to investigate more to see if there's something that's going on with introgression and outcrossing with other species, if that's also confounding our values to give us really high values in our time divergence. Going for population structure, we can. We saw that in our PCA plot, the admixture and the phylogeny, they were identical to the results that the PCA gave us. But those are available if you want to see them during the question time. As integration played a role in domestication, we're actually not sure because what we saw in the divergence time was unexpected, and in geological timescales, more sequencing analysis would have to be done in order to kind of get to the bottom of that. And as far as the genetic basis of flowering, we actually did see some potential genes with some SVs involved, which is pretty cool. So as far as the future work, I want to create a scaffolded genome in order to identify the sex chromosomes, and also sequence a type B avocado that is also highly scaffolded. We also want to investigate different individuals, Persea to see if there's any more evidence of integration in Mexican avocados, and use the Pacado hi-fi so that we can do better SV calls. Also, I use my computer science background for machine learning and rapid SNP chip prototyping for genotyping such as ecotype and sex. And I'm currently talking to a faculty member in the UC for looking at plasticity and seasonal flowering of avocados. So I want to thank my PhD lab, Brian Cott Lab at UC Irvine, our Paya Lab in Riverside, and Cantu Lab in Davis, which are our collaborators, as well as our current lab at the Rossavara Lab. So If you have any questions, I'm ready now to take this.